Hey, it's Mr. Shrum, and I'm back to finish this genetic mutations tutorial. And we were just about to talk about mutations. Mutations, if you've ever seen X-Men, right? Mutants. Why do they call them mutants? These are people with changes or differences than normal, what would you say, neurotypical, genetic normal humans. And that's because they have mutations. Now that show and those movies are exaggerated, but it's all based around this genetic mutation. And that is a change in the genome. Mutations add to the genetic variation of a species and can lead to the development of new traits. Genetic mutations occur in all organisms. Even species that reproduce asexually, which means they can just reproduce themselves, can develop mutations in their DNA over time. Many mutations are random, but chemicals and other substances in the environment can also cause these mutations. Substances that trigger mutations in genes are called mutagens. Geneticists, scientists that study genetics, categorize mutations into several types, depending on the type of change that occurs in the DNA sequence. And we're going to examine the different ways in which a strand of DNA can mutate. Here are the types of mutations. We have substitution. That's when one nucleotide is substituted for another. A substitution may change a codon and the resulting amino acid. It may cause a silent mutation in which there is a change in the codon, but the resulting amino acid is the same. It may also convert a codon to a stop codon. We have deletion. That is when a mutation removes a nucleotide from the base sequence. Deletions may be as small as one or a few base pairs, or they may result in removal of complete genes entirely. Duplication, that is when a portion of DNA duplicates. It makes a copy of itself one or more times. Duplication may result in multiple copies of genes, which may alter the structure or function of the resulting function, or the resulting protein. Insertion, that is when a mutation involves the addition of one or more bases to the sequence. Insertions may disrupt certain genes, or they cause alterations in the structure or function of resulting proteins. Inversion, that is when a portion of DNA reverses itself, and that reversal may lead to the disruption of genes. So we see how the C and D have been switched or inverted. And then finally, the last one is translocation. That is a mutation in which sections of DNA from two different chromosomes swap locations. A translocation may change the chromosomal location of genes. Okay, so identify the types of mutation represented by the change in each DNA sequence. So we have ATGC, and the G is deleted, that's deletion. We have ATGC moving to ATTC, that is a substitution. The G is substituted out for T. ATTCGC, ATTCGGC, they have inserted a G between the C and G here. ACGT to TGCA, inverted, they just flipped all the letters backwards. ATTG, ATTG, ATTG. That is a duplication. That's just ATTG repeated twice. And then we have ATTC, which is one chromosome, and the second chromosome, CGATTC. And that changes from chromosome 1 to chromosome 1 in ATCGA and chromosome 2, TCTTC. So that is translocation. Inherited mutations come from one or both parents via the sperm or the egg cell. Once inherited, these mutations exist in all the cells in the body. Let's look at some disorders caused by inherited mutations. We have phenylketonuria, or PKU, and that is caused by a mutation on the PAH gene. That gene codes the enzyme that breaks down the amino acid phenylalanine. The increased levels of phenylalanine in the blood can cause mild to severe brain damage. Other effects include delayed development, skin disorders, behavioral problems. PKU is an autosomal recessive inheritable condition. A child may be affected by PKU only if the child inherits 2 copies of the defective gene, one from each parent. You can see the chromosome and the mutation here. Breast cancer. Mutations in certain genes that cause breast cancers, such as BRCA1, BRCA2, show hereditary inheritance. And the pattern of inheritance is autosomal dominant. A person's chance of developing breast cancer can increase by inheriting just 1 copy of the mutated gene from either parent. These genes encode proteins that play a role in repairing DNA damage. Mutation in these genes hamper the DNA repair mechanism and may lead to cancer. Cystic fibrosis. This occurs because of a mutation on the CFTR gene. Cystic fibrosis is inherited in an autosomal recessive pattern. That is, both copies of the gene must be mutated. The CFTR gene codes a protein responsible for salt and water transport across cells. The mutated gene produces a defective protein that causes a buildup of thick, sticky mucus that can affect several organs. It can block airways, leading to severe coughing and wheezing that damages the lungs. The mucus can cause blockages in different digestive and reproductive organs, which leads to inefficient functioning of these organs. Somatic mutations are mutations that occur in the somatic cells of an organism. Somatic cells are all the cells that make up the body of an organism. These cells do not divide to form sex cells or gametes. Unlike inherited mutations, somatic mutations are not present in an individual from birth. They are acquired through exposure to mutagens during the individual's lifetime. These acquired mutations are not passed on to offspring. A somatic mutation will be present only in the type of cell in which the mutation has occurred. It does not exist in all cells of the body. For example, consider a skin cell that mutates because of exposure to ultraviolet light. The exposure leads to a cancerous change in the cell. All daughter cells of this mutated skin cell will have the mutation, but other cells of the body will not show this mutation. Most cancers arise from somatic mutations. Here is a mole with an irregular border, and that can be an indication of skin cancer, which is caused by somatic mutation. So what are the effects of mutations? They have a reputation for being harmful, and some definitely are harmful to organisms, but most mutations don't affect our survival at all. While rare, some mutations can even lead to beneficial traits. Neutral mutations have no effect, positive or negative, on an organism's survival. They're kind of just there. They may not even be expressed in the organism. Others, though expressed, will not be beneficial or harmful to the individual. Some color mutations are neutral. For example, some animals may exhibit a slight variation in color because of a genetic mutation that causes an increase or decrease of pigment in their coloring. If the color variation doesn't give the animal a better or worse chance at attracting a mate, the ability to camouflage itself from predators or the success of reproducing it is a neutral mutation. So we have neutral, we have harmful mutations cause changes in the genome that decrease the species' chance of survival. These type of mutations lead to genetic diseases, usually. Some color mutations are harmful as well. In some grasshoppers, a mutation in the gene responsible for pigmentation turns them pink. Pink grasshoppers can't blend into their environment as well as green or brown grasshoppers can. And predators can easily spot pink grasshoppers, which results in a lower chance of survival. Beneficial mutations. Certain mutations give organisms attributes that increase their chance of survival. The process of natural selection favors such mutations, which then spread through the species. A well-known example is seen in peppered moths. Speckled peppered moths can camouflage themselves on lichens that grow on trees. During the Industrial Revolution, smoke from factories turned these trees black and killed the lichens. Predators could easily see the peppered moths. However, a mutation caused some moths to have black pigmentation. These black moths were able to blend in with the blackened tree trunks. They escaped predation, survived, and reproduced. This mutation spread in the species. For a while, all moths in the area were black. Mutations are an important mechanism for introducing genetic diversity in asexually reproducing organisms. Asexual reproduction involves just one parent producing copies of itself. All offspring have the same genetic material as the parent. There is no mixing or recombination of genetic material. So as a result, there's no variation in genes from generation to generation. Spontaneous mutations can add to diversity of a genetic pool of species that reproduce asexually. A well-known example is the methicillin-resistant, or the MRSA, MRSA. This bacterial strain has emerged from a number of mutations that make the bacteria resistant to antibiotics. You can see that here, where antibiotic cannot damage a cell wall. And I'll leave off here for now, and we'll finish up next time. But until then, have a great rest of your day, and see you soon. Bye-bye.